Hello students and my dear friends. In this session, we'll discuss some of the important microbiology MCQs which will be helpful in the lab technician recruitment examination. First question, Louis Pasteur is not associated with option A, introduction of complex media, option B, discovery of rabies vaccine, option C, discovery of Mycobacterium tuberculosis, option D, disproved theory of spontaneous generation. Theory of spontaneous generation, it is also called as theory of abiogenesis. Here, the right answer is option C, discovery of Mycobacterium tuberculosis. So Mycobacterium tuberculosis, it was discovered by the scientist Robert Koch. So here Louis Pasteur, he did not discover the Mycobacterium tuberculosis. So what are other contributions of Louis Pasteur? He used or he prepared the culture media, and he was the first person who prepared the vaccine for the rabies, and he disproved the theory of spontaneous generation, and he introduced the techniques of sterilization, and he established the different growth need of different bacteria. So that is helped in the complex media, and he developed the live vaccines. Next question, microscope was invented by option A, Ronald Ross, option B, Robert Koch, option C, Antony van Leeuwenhoek, option D, Louis Pasteur. Here, the right answer is option C, that is Antony van Leeuwenhoek. Antony van Leeuwenhoek, he was the first person who prepared the microscope. Okay, he has the hobby of grinding the lenses, and he used to prepare the new lenses. So with his lenses, he prepared the first microscope. With his microscopes only, he observed the various forms of the microorganisms. Now what we are known as rod-shaped bacteria as well as protozoa. So he observed under his microscope. Next question, Treponema pallidum was discovered by option A, Robert Koch, option B, Ford, option C, Schwade and Hoffman, and option D, Hansen. Here, the right answer is Schwade and Hoffman. So these two scientists, they discovered this Treponema pallidum. Treponema pallidum is the spirochete. It is a causative agent of the syphilis. They discovered in the year 1905. And if you see other options, Robert Koch, he discovered Mycobacterium tuberculosis, Vibrio cholera. And if you see Twat, he discovered the bacteria of H. Hansen, he discovered the Mycobacterium leprae. Next question, dark ground microscopy is used for detection of option A, spirochetes, option B, chlamydia, option C, fungi, option D, virus. Here the right answer is option A, spirochetes. So spirochetes, they are discovered by the dark ground microscopy. And if you see fungi, generally cannot be seen by the microscopy. They need electron microscope. And viruses, they can be seen under electron microscope. But dark ground microscopy, it is used for detection of the spirochetes. Next question, dye used in fluorescent microscopy. Option A, thioflavin T, option B, Congo red, option C, brilliant blue, option D, auramine. Here the right answer is option D, auramine. So here, what is meant by fluorescent microscope? So fluorescent microscope, it helps in the rapid diagnosis of infections. So here, these fluorescent dyes, whenever they are exposed to the ultraviolet light, these fluorescent lights, they absorb the energy and they emit the visible light, which means they fluorescence. So tissues or organisms, they are stained with these dyes and examined with the UV light in a special adapted microscopes, which are seen as fluorescent objects. So in this technique, antibodies also can be labeled with the fluorescent dyes. The antibodies can be coated with the fluorescent dyes, and this fluorescent dyes, it react with the homologous antigen at the cell surface, that is called as direct immunofluorescence test. So in this fluorescence test, so generally we use fluorescent dyes such as aramine, rhodamine, and lysamine, and fluorescence isothiocyanate are used. So in these options, aramine is the fluorescent dye. Next question. Silver impregnation technique is used in the identification of option A, spirochetes, option B, leptospira, and option C, Borrelia, and option D, all of the above. Here, the right answer is option D, all of the above. So silver impregnation, it is used to identify the spirochetes. If you see spirochetes, they include Treponema, Borrelia, and Leptospira. So here the answer is like spirochetes. All the spirochetes can be identified by this silver impregnation methods. And what are the other techniques used for identification of spirochetes? Like even dark ground microscopy and immunofluorescent techniques. They are also helpful in the identification of the spirochetes. Next question, non-motile organism. Which of the following is non-motile organism? Option A, E. coli. Option B, Vibrio cholera. Option C, Proteus mirabilis. Option D, Shigella dysentery. Here the right answer is option D, that is Shigella dysentery. Shigella dysentery is non-motile organism because they do not possess the flagella. If you see E. coli, so E. coli, they have the peritrichous flagella, so they show the motility. Vibrio cholera, it has single polar flagella, it shows the vibratory motility or dotting motility. And Proteus mirabilis, it is highly motile organism. So the answer is Shigella dysentery. Shigella dysentery is non-motile organism. Next question. Mesosomes in bacteria are functional unit for option A, lipid storage, option B, protein synthesis, option C, respiratory enzymes, option D, none. Here, the right answer is option C, respiratory enzymes. So if you see mesosomes, they are also called as chondroids. Mesosomes are formed by the invaginations of the plasma membrane. And see, mesosomes are more prominent in the gram-positive bacteria, and they are the principal sites of the respiratory enzymes. And they are analogous to mitochondria of the eukaryotes. In case of eukaryotes, mitochondria helps for the respiration. But in case as bacteria is prokaryote, so there is no mitochondria. So instead of mitochondria, mesosomes, they took up the respiratory function. Here, the right answer is respiratory enzymes. Next question. Which of the following is not capsulated? Option A, pneumococcus. Option B, cryptococcus. Option C, meningococcus or meningococcus. Option D, proteus. Here the right answer is proteus. So proteus bacteria is non-capsulated. So what is a capsule? So capsule is a outer viscous layer which is secreted around the cell wall, outer gelatinous layer. So here if you see pneumococcus, that is Streptococcus pneumonia, possess the capsule, and Cryptococcus neoformans, it is a capsulated fungi, and Neisseria meningitidis also possess the capsule. So here the right answer is proteus. Proteus is non-capsulated organism. Next question. Which of the following pathogen does not satisfy Koch postulates? Option A, Bacillus anthrax. Option B, Mycobacterium tuberculosis. Option C, Clostridium tetani. And option D, Lepra bacilli. Here, the right answer is option D, Lepra bacillus. So Lepra bacilli, they do not follow the Koch postulates. So what are Koch postulates? Association, isolation, inoculation, re-isolation. So here, if any bacterium follow the Koch postulates, then only it is considered as a pathogenic organism. If the bacterium, if it doesn't follow the Koch postulates, then it is considered as a non-pathogenic organism. But if you see Mycobacterium leprae, so even though it is the a pathogenic organism, it doesn't follow the Koch postulates. So this is the only organism it doesn't follow the Koch postulates, but still it is considered as a pathogenic organism. Next question. Correct sequence of bacterial growth curve. Option A, log phase, lag phase, stationary phase, decline phase. Option B, lag phase, log phase, stationary phase, decline phase. Option C, stationary phase, lag phase, log phase, decline phase. Option D, lag phase, exponential phase, log phase, death phase. Here the right answer is option B, that is lag phase, log phase, stationary phase, and a decline phase. So here, the bacterial growth curve. Okay, so the one bacteria, it is divided into two daughter cells. That process is called as a binary fission. So there are four phases are there in the bacterial growth curve. The first phase is lag phase. So lag phase is the longest phase, and in this phase, there is no increase in the cell number. So just in the lag phase, the bacteria try to adjust to the new environment. And in the second phase, that is log or exponential growth phase. Log phase, it is also called as exponential growth phase. So in this phase, the cells actively multiply and they reaches to the maximum number. And third phase is the stationary phase. So stationary phase, the number of cell birth is equal to the number of cell death. So there is a equilibrium. And in the decline phase, so here the cells starts dying. So here the population of the bacteria decreases in the decline phase or death phase. Next question. An antiseptic means option A, an agent applied on the skin to eradicate pathogenic microbes. Option B, used to sterilize inanimate objects. Option C, an agent which kills only bacteria but not the spores. Option D, kills all microorganisms. Here, the right answer is option A, an agent that is applied on the skin to eradicate the pathogenic microbes. So here, disinfectant. So disinfectant is also chemical substance which kills the microorganism. And antiseptic is also chemical substance only. It also kills the microorganism. But what is the difference? Here, antiseptic, it can be applied safely on the living tissue. It can be safely applied on the skin. So here, if you see antiseptics, they can be applied on the external surface. And so here, antiseptics, they are not used to sterilize inanimate objects. They are used by the disinfectants. And here, the right answer is an agent that applied on the skin to eradicate the pathogenic microbes is called as the antiseptic. Next question. The best skin disinfectant is option A, alcohol. Option B, betadine. Option C, Savlan. Option D, phenol. Here the right answer is betadine. So here, if you see, all these options are the skin disinfectants only. So here, what is the skin disinfectants? Antiseptics, they are also called as the skin disinfectants. So in the previous slide, we discussed what is antiseptic. So antiseptic that can be safely applied on the living tissue. Okay. So here, antiseptics, they are also known as skin disinfectant. If you see alcohol, betadine, Savlan, so they are used as skin disinfectants. But if you see the best, the question is the best skin disinfectant. Here the answer is betadine, that is a povidone iodine. Why povidone iodine is best skin disinfectant? Because povidone iodine is less irritant and it causes the less staining. So that's what betadine is a best skin disinfectant. Next question. Which of the following is true about pasteurization? Option A, it kills all bacteria and spores. Option B, it kills all bacteria except thermoduric bacteria. Option C, it kills 95% of microorganisms. Option D, all bacteria are destroyed. Here, the right answer is option B, it kills all bacteria except thermoduric bacteria. So here, what is meant by pasteurization? It is the sterilization of the milk. So by using of the pasteurization, we can kill only the vegetative forms of the bacteria, but not the spores. So here there are two methods are there, holder method and flash method. In the holder method, we heat the milk at 63 degrees centigrade for 30 minutes. In the flash method, the milk it is heated at 72 degrees centigrade for 15 to 20 seconds. So here by this, generally we kills all the forms of the vegetative bacteria, but not the spores. But if you see bacteria such as Mycobacterium tuberculosis and Brucella and Salmonella, they are killed except Coxiella burnetii. So here Coxiella burnetii, it is a thermodiuric bacteria. So the right answer is option B. It kills all the bacteria except thermodiuric bacteria. Next question. Moist heat kills of the following except option A, Brucella, option B, Salmonella, option C, Mycobacteria, option D, Coxiella burnetii. So as we discussed in the previous slide, the right answer is Coxiella burnetii. So here Coxiella burnetii, it doesn't killed by the moist heat. So just now we discussed pasteurization. Pasteurization is also one of the moist heat method of sterilization. So as you know, heat sterilization is done by either dry heat or moist heat. So pasteurization is the sterilization by the moist heat. So here with this pasteurization, as we discussed, Mycobacteria, Brucella, Salmonella are killed. This Coxiella burnetii is not killed. Next question. Tyndallization is a type of option A, intermittent sterilization, option B, boiling, option C, pasteurization, option D, autoclaving. So here the right answer is option A, intermittent sterilization. So what is tyndallization? The media which consists of sugar or gelatin are sterilized by heating at 100 degrees centigrade for 20 minutes on 3 consecutive days. So when we heat this culture media on first day, so it kills the vegetative forms of the bacteria. So next day, if there are any spores are present in that culture media, next will germinate. Again, whenever we heat on the second day, that is at 100 degrees centigrade for 20 minutes, on second day, that vegetative forms of the bacteria are killed. And if there are any spores are remnant, so they will germinate on day 3, and again day 3 also we heat them at 100 degrees centigrade for 20 minutes, so that these spores they germinate and they are killed by using of this tyndallization. So tyndallization, it is given by the scientist John Tyndall. Next question. 40% of formalin is used to sterilize. Option A, plastic syringes. Option B, clothes. Option C, all microbes and spores. Option D, stitches. Here, the right answer is option C, all microbes and spores. If you see formalin, so formalin is nothing but the formalin, it consists of 35% solution of formaldehyde. So formaldehyde and glutaraldehyde, they comes under aldehydes. So formaldehyde is bactericidal, viricidal, and sporicidal. So as you know, formaldehyde, it available in the two forms. Formalin, that is solution, 35% solution of formaldehyde. And it is also available in the formaldehyde gas. So this formaldehyde is used for sterilization of instruments and heat sensitive catheters, fumigation of wards, sick rooms, operation theaters, laboratories, and disinfection of clothing, bedding, furniture, and books. So here, the right answer is option C. Next question, percentage of glutaraldehyde used. Option A, 1%, option B, 2%, option C, 3%, option D, 4%. So here the right answer is option B, that is 2%. 2% glutaraldehyde, that is Sidex. It is an aldehyde agent. So it has a broad spectrum action. This glutaraldehyde is effective against bacteria, fungi, viruses, and even it show action on the spores. It is also one of the high-level disinfectant solution. Next question, all are sporicidal agents except option A, formaldehyde, option B, glutaraldehyde, option C, ethylene oxide, and option D, isopropyl alcohol. Here the right answer is isopropyl alcohol. So isopropyl alcohol is the what is the sporicidal first of all? So sporicidal, it is a agent which kills the spores. So here if you see options such as formaldehyde, glutaraldehyde, and ethylene oxides, they are the sporicidal agents. They kill the like spores. But if you see this isopropyl alcohol, so isopropyl alcohol, it doesn't kill the spores. It kills only the like vegetative forms of the like microorganisms. Next question: bacterial spores are best destroyed by option A, UV rays, option B, autoclaving at 121 degrees centigrade for 20 minutes, 15 to 20 minutes, and option C, hot air oven, option D, infrared rays. Here the right answer is option B, autoclaving at 121 degrees centigrade for 20 minutes. It is at 15 lb pressure for 15 to 20 minutes. So here, if you see other options, they also kills the like spores. Okay, so this except this infrared rays, but here the question is the spores are best destroyed. So here they are best destroyed by using a autoclave. Thank you.